The adenylate cyclase pathway is one of several mechanisms which cells use to convert a chemical signal on the outside of a cell into a change in behavior inside the cell. In this guided exploration, we will step through how this signal transduction process occurs and how these inputs are integrated and regulated by intracellular pathways. The triggering event is the arrival of stimulatory signaling molecules, for which the cognate receptors in this signal transduction pathway are present on the cell surface. These molecules bind to each other with extremely high affinity, such that concentrations as little as 10 to the minus 9 molar can be detected from the extracellular milieu. Although there are many different receptors, each of which specifically bind only one of the many types of hormones and other extracellular signals, all receptors which regulate the adenylate cyclase pathway belong to a common family. All members of the family share a common topology, consisting of 7 consecutive transmembrane helices. The only member of this family whose structure is known is bacteriorhodopsin, although it is not a receptor. The binding of a ligand causes the receptor to change conformation. The nature of this conformation is unknown, but it in turn affects the binding of the heterotrimeric G protein GS to the intracellular side of the receptor. In its resting state, GS is a complex of 3 subunits, alpha, beta, and gamma, in which the alpha subunit binds GDP. The alpha and gamma subunits are each anchored to the cytosolic side of the membrane by myristoyl groups. The activated receptor stimulates the G protein's alpha subunit to exchange the bound GDP for GTP. The binding of GTP in turn causes the alpha subunit to dissociate from the beta-gamma portion of the G protein and to release the receptor. Consequently, the receptor can go on to activate several other G proteins before it eventually releases its bound hormone and returns to the inactive state. The now active alpha subunit of GS in turn activates the membrane-bound enzyme Adenylate Cyclase to catalyze the synthesis of cyclic AMP from ATP. This results in the buildup of intracellular concentration of this second messenger. The stimulatory activity of GS-alpha is self-regulating. In addition to its ability to stimulate adenylate cyclase, GS-alpha is a GTP ACE that eventually hydrolyzes the bound GTP back to GDP. When this occurs, the alpha subunit rebinds the beta and gamma subunits, thereby returning GS to its off state. In addition to the self-limiting activity of GS, cyclic AMP is continuously degraded by a specific phosphodiesterase. Therefore, in the absence of stimulatory signals, cyclic AMP concentrations rapidly return to their resting levels. Also, some hormone signals result in the activation of G proteins that inhibit adenylate cyclase. Therefore, the intracellular concentration of cyclic AMP can integrate a variety of positive and negative extracellular signals into a single second messenger readout. But how do changes in the concentration of cyclic AMP result in changes in cell function? The answer is that cyclic AMP in turn activates an enzyme known as cyclic AMP-dependent protein kinase. Cyclic AMP-dependent protein kinase is a tetrameric protein that consists of 2 catalytic subunits and 2 regulatory subunits. In the resting state, it is inactive, because the regulatory subunits prevent the catalytic subunits from functioning. However, when intracellular cyclic AMP levels rise, 2 molecules of cyclic AMP bind to each subunit of the regulatory dimer, which causes the dissociation of the catalytic subunits, thereby freeing them to act. The catalytic subunits then catalyze the ATP-dependent phosphorylation of specific serine and threonine residues on proteins that participate in such pathways as glycogen metabolism and gluconeogenesis, thereby altering the activities of these enzymes. Of course, in the yin and yang of cellular regulation, there is also a corresponding mechanism to counter the effects of phosphorylation by cyclic AMP-dependent protein kinase. This is provided by cellular phosphatases, enzymes which hydrolyze away specific phosphate groups on certain proteins. These phosphatases are in turn regulated by other pathways, but that is another story.